That's recording okay. So we'll just move to the slides. Okay, can you all see the slides now?
Yes.
Yes.
Fine. So this is the last part of the BIOS 3032 course. There'll be a practical next week. You'll do with Dr. Jan over in the NCMH laboratories. That'll be next Wednesday afternoon. And if you are watching a recording of this, then it's strongly recommended that before this short lecture, you see the first 7 minutes 50 seconds of the polyacrylamide gel electrophoresis and ultracentrifugation video by Professor Tom Lowy and colleagues. It's on the NCMH Outreach BIOS 3032 page, YouTube page. You can find a link on there. And as far as the course is concerned, we've already considered molecular weight measurement and average molecular weights, techniques like osmotic pressure or sedimentation, equilibrium. You need to know one absolute method for molecular weight measurement in this course, which doesn't require calibration standards, and we've done that with osmotic pressure, which gives average molecular weight. Familiarity or the knowledge that there is this technique called sedimentation equilibrium in the ultracentrifuge, which can measure the average molecular weight, also an absolute molecular weight technique, not requiring standards. But you're required to know about a technique which is known as a relative molecular weight technique. That is, it gives the molecular weight of a macromolecule relative to standards. And this is polyacrylamide gel electrophoresis, and it refers to protein molecules, the measurement of molecular weight of proteins, again, relative to standards. So the measurement of the molecular weights of proteins relative to protein standards using this technique. And these are the sort of patterns that you get. And in many, if you look at a biochemical, biological science, scientific journal and look and see some of the papers inside the journal, then there's a very good chance you'll see an example or examples of these types of what's called SDS page patterns illustrating protein structure, protein composition, and molecular weights. It's a technique which has revolutionized biochemistry, biological science over the last 50 years or so. Slaterophoresis, electrophoretic mobility, is simply the movement of a charged macromolecule under the influence of an electrical field. So if you have a solution of molecules, macromolecules, and they possess charges, proteins, especially all proteins possess charge through the charged amino acid groups. Of course, the end residues will be charged, the amino end and the carboxylic acid end will be charged, so long as the pH is not too acidic or not too alkali, then both ends will be charged. But also, if you have amino acids like aspartic acid, glutamic acid present, they give a negative charge, or lysine, arginine, they give, histidine, they give a positive charge. So you tend to find that protein solutions are charged, and depending on whether the pH is below or above the isoelectric pH, the protein will have either a net positive, it's below the isoelectric pH, or net negative if it's above the isoelectric pH value charge. And if it's actually at the isoelectric point, if the conditions, buffered conditions, are such that we're at the isoelectric point, then it won't be, it'll still be charged, but the numbers of positive charges equal the numbers of negative charges. If you've got net charge on the macromolecule of protein, or you could have a nucleic acid, which has got lots of negative groups due to the phosphodiester, the phosphate carbohydrate backbone, then that will have a large negative charge that will move under the influence of electric field. Or many polysaccharides have charges, either negative charges such as alginates and pectins, or positive charges like chitosanes, and they will also move under the influence of an electrical field. Slaterophoresis is used for small-scale purification. In the protein world, it's for determining subunit composition of proteins and for determining the relative molecular weights relatives to standard molecules of known molecular weight. And this isn't a SDS page setup. This is just a basic electrophoresis setup taken from the Firth and Moore book, just to illustrate the general principles of electrophoresis. So let's say we put our solution of let's say protein in this sample well, and then we have an electrical circuit across this setup here. This end is connected to the anode, so this is positive end, and this end is connected to the cathode, and this is the negative end. So if we have a solution of protein here and we switch on the field, then the protein molecules will move. If it's got a net positive charge on the protein, it will move towards the cathode. We call these cations because they're moving towards the cathode. Or if it's got a net negative charge, it'll move towards the positive anode. So these are called anions. They move towards the positive anode if they have a negative charge. And the reason for having a gel is that if you switch the electric field off, then if it's a solution, then everything will just jumble up again through, Brownian motion. And again, second law of thermodynamics, everything will be randomized. But if the sample solution, the molecules have moved through a gel, then the gel will tend to preserve that separation, at least for a period of time, and hinder the diffusion back in. That's why we use a gel. But if we want to use this sort of setup to measure the molecular weights of proteins, we've got some problems, because the how fast the protein moves on the gel depends not only on the molecular weight. The larger the molecular weight, then the slower the molecules will move because they will be hindered by the gel particles. But the rate of electrophoretic movement will depend on the charge. So the more charge the protein has, depending on the numbers of aspartic glutamic acid groups, arginine, lysine residues, then the greater the net charge, whether it be negative or positive, then the faster it will move. So it won't just be molecular weight that dictates how fast the proteins will move. And also, proteins have different conformations. So if you've got a more extended conformation of a protein molecule, that will also go slower. So if we want to use this sort of technique for measuring the molecular weights of proteins, we have to get around the fact that you have variable charges on protein molecules, and also you have variable conformations. And SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis gets around this problem in a very clever sort of way. So the first thing we do is we bind to the proteins a molecule called dodecyl sulfate, or we mix the protein with sodium dodecyl sulfate. And if we expose the protein molecule to an excess, that is over 1% of this basic detergent called SDS, as described in the Tom Lowy video, then we find that 1.4 gram of SDS binds per gram of protein. And the SDS molecule has a negative charge, so you find that if you bind SDS to the protein with SDS in excess, you tend to find one negative charge through the SDS binding per 2 peptide bonds. To make this work, you have to also, first of all, disrupt any disulfide bonds that form between cysteine residues in the protein molecule. And we've talked about cysteine before. This is the SH group, and the SH groups come together to give disulfide bonds. So you need to, first of all, break the disulfide bonds by using thiol reducing agents. And again, this is referred to in the Tom Lowy video. Thiol reducing agents such as mercaptoethanol and di-thioethol. Now, these are quite nasty chemicals. They don't smell very nice as well, so you have to be quite careful when using these thiol reducing agents, and this is normally done in a fume cupboard. So you break the disulfide bonds, and then you bind the SDS to the protein, and that gives one negative charge per 2 peptide bonds. So you confer on the protein molecule a very high and uniform negative charge, and this swamps out any small variations between proteins due to the different arginine, lysine compositions, or different aspartic acid, glutamic acid compositions. You have this swamping one negative charge per 2 peptide bonds caused by the binding of the SDS. So you confer on the protein a more or less uniform negative charge per unit length across the protein. And because the molecule is very heavily negative, negative charges repel, and so the previously globular conformation is denatured, and then the proteins adopt an extended conformation, a sort of flexible rod type of conformation. So proteins end up with a uniform charge per unit length, with some small variation depending on the aspartic acids and things present, but by and large it's a uniform charge per unit length, and also they'll have the same conformation, this extended rod shaped conformation. And since the length of the proteins will differ only in their length, having the same charge per unit length ratio, and since the length of a protein will depend on the number of peptide bonds, then the length will be proportional to the molecular weight. So separation then is, the proteins will differ in terms of the separations based only on the molecular weight. You can see from this diagram here, this represents the polyacrylamide gel particles, and the smaller proteins will get through easier compared to the larger and very large molecules. So it's a very nice way of separating proteins of different molecular weight. And also we can measure the molecular weights, because the mobility in the gel, or through the retarding, I mean slowing down gel, relative to a marker or tracer dye, again, which the video referred to, will depend on the charge per unit length, which is a constant, and divided by the log of the molecular weight. And we put a little symbol R here because it shows it's going to be relative to standards. So it's been found empirically that the mobility on a gel under the influence of an electrical field will depend on the charge. This means proportional to charge per unit length, which is a constant, divided by the log of the molecular weight. Or the mobility is proportional to the reciprocal over the log of the molecular weight. And over relatively small molecular weight ranges, the sort we do with proteins, then this this relation here can approximate to D is proportional to minus log 10 m. So you plot the log of molecular weight against the relative mobility relative to the marker or tracer dye, then you get a straight line with negative slope. So the idea is you define this line using proteins of known molecular weight. You run them on your gel and you produce this calibration plot. Then you measure the mobility of your unknown material and then read off on this logarithmic graph here what the log of the molecular weight is, and then take the anti-log, and that gives you the molecular weight. So it's very simple in how you measure molecular weight of proteins with this technique. The relative mobility D is sometimes written as RF. Okay, that's shown here. So this is the setup that was described in the video. So generally speaking, you have your standards. You run the standards at the same time as you run your unknowns. So this is the retarding polyacrylamide gel with these little inlets here where you inject your samples or different samples, and also you have at least one lane which contains your standard proteins of known molecular weights. So the anode is here, that links to the positive electrode on the battery or whatever power pack, and this links to the negative electrode or negative terminal on the power pack or battery, and then you load your samples and you switch on. You detect your protein by dyeing the gel or the protein with a blue marker dye called Kumasi blue. So that will detect where the protein is on the gel. So use Kumasi blue to pick up the position on the gel. These are the patterns that you get. These are this is the standard lane here in proteins of known molecular weight, and these are samples containing snake venom toxins, and this is comparison of differences between an adult snake and a newborn snake. This is a known toxin called crotamine from snake venom, and you can see the molecular weight of this is something between 2.5 and 6.2 based on these standards here. From the standards, you can produce the calibration plot of relative mobility, range from 0 to 1. That's the mobility of a tracer marker dye. And the smaller the protein, the faster it will sediment. This is bovine serum albumin, molecular weight's about 67,000. This is egg albumin, 45,000, and smaller proteins here. So you form your calibration plot, and then you measure your relative mobility on the gel. Then you read off from this plot here what the molecular weight is. Now, either you can plot log molecular weight against mobility, either as D or F, or you can plot the molecular weight on a logarithmic axis. And that means you can read off the molecular weights directly. You can spot a logarithmic axis because the spaces between the numbers are not uniform. But beware when you do this. So on this axis here, say if you read off what the molecular weight is, let's say bovine serum albumin, that comes out to be about 67,000. But on this axis here, it says molecular weight times 10 to the minus 3, 60. And that means you don't multiply that by 10 to the minus 3. It means that these numbers on this axis are molecular weight values that have been multiplied by 10 to the power minus 3. That means to get the correct molecular weight, you need to multiply by 10 to the power 3. Okay, so that's what that means. And why this is relevant is, I think it's question 7 on the assessed questions that I've given you. You have a similar sort of plot to deal with. So you don't multiply by 10 to the minus 3 these numbers. You have to multiply by 10 to the power 3 to get the actual numbers. Otherwise, you get nonsensical molecular weight values. You can't get molecular weights less than 1. So always make sure when you find something that your answer makes sense. A couple of points here. This is for proteins. So it doesn't work for polysaccharides. SDS don't think binds to polysaccharides, or certainly not in a uniform sort of way. So it doesn't work for polysaccharides. With DNA, you use a different type of gel, a more porous gel, because DNA molecules are larger. So we use agarose rather than polyacrylamide, and also because DNA molecules already have a regular charge because of the phosphate residues on the sugar phosphate backbone. They have a uniform negative charge across the DNA molecule. There's no need to bind any sort of negative charge like SDS things to the DNA, and also use different types of stain. We tend to use things like ethidium bromide. So for DNA electrophoresis, it's different. You don't use SDS, and we use a different type of gel. I think that's relevant to the DNA question we've given you on the assess questions. I forget which question number that was. Okay, so bear that in mind. And also, there's two other things you need to take into consideration when using this technique for proteins. If you have some proteins have already a very high charge, and one example is histones. And histones are used in eukaryotes as a way of compacting the DNA. They have very high positive charges to compact the negative charge DNA. When you have a very high positively charged protein like histidine, then even binding one SDS or one negative charge per two peptide bonds, that's not enough to swamp out the local charges of the histidine molecules. That's an example where SDS page doesn't work. And also, if you have a multi-subunit protein in which the subunits are non-covalently linked together, for example, hemoglobin is a tetramer. Now, the molecular weight of a hemoglobin tetramer is about 64,000. But if you measure the molecular weight of hemoglobin with SDS page, you get a value of about 16,000. And the reason for that is that the SDS, besides the high negative charge, besides giving proteins a uniform conformation, rod shape, because of the repulsive effects of the negative charges will also break the molecule up into its subunits. So that's why you get a molecular weight of 16,000 for hemoglobin. It's a technique for giving the subunit molecular weight of proteins. And unless you've got proteins which are linked together by the disulfide bonds, but then if you added mercaptoethanol or DTT, you would have disrupted that. So it gives the subunit structure or subunit molecular weights of protein molecules. And that can be a good thing because we have a technique for giving the subunit molecular weights of proteins. If you use that in conjunction with a technique which gives the molecular weight of the intact protein molecule, like osmotic pressure or sedimentation equilibrium in the ultracentrifuge, you could use the two techniques together to work out the subunit composition of the protein molecule. So let's just come out of that. Good. Are there any questions on that? You should be able to answer the two electrophoresis questions in the assess questions that you have been given. And if you use this in conjunction with the Tom Lowy video, which, if you're watching this as a recording, it's in the same group of YouTube recordings for the rest of this course. So if you use this in conjunction with the Tom Lowry video and also the notes in the Bertha Moore Self-Assembly of Macromolecules book, which I've given you, I think a hard copy or online, you should be okay. All right.